Well, you saw Drew going down for a dive earlier today to have a look at the benthic ecosystem here at Cape Evans. But you've actually been looking at something else over the other side of the Ross Sea. So whereabouts was that?
Yep, we went over to a place called Granite Harbour, and yeah, we stayed there for about 2 weeks doing experiments under the ice.
Cool, and so can you tell us about that?
Yeah, it was all this benthic chamber works. So Granite Harbour does have a variety of different habitat types. It's got rocky bits and sandy bits, and for this experiment we chose a patch of sand. It's about 14 or 15 meters deep, and it's sort of a homogeneous patch of sand. And these are benthic chambers, and we had 16 of these little units, these chamber units down on the seabed, and they're about 50 centimeters by 50 centimeters square. And what we do with these is that the chambers have aluminium sides, the base there, that we press down into the sediment, and that encloses a certain area of seafloor. And then you put this acrylic lid over the top of it, and that seals it so that it's basically a sealed package of water, that there's no water flow into and out of the chamber. And then we measure the change in the water chemistry over time to see what's happening with the benthic food web. And one of the things that we're really interested in is the benthic algae, the ice algae that grows on the underside of the sea ice. And so that stuff falls down to the seabed, and that's a really important source of food for the animals living on the seafloor. And so what we did is we actually collected under ice algae, we concentrated it, and we squirted it into some of these chambers and not others. And then we looked at changes in the utilization of that food in the different chambers. Also, the chambers had different numbers of animals. We used brittle stars as our experimental animal, and there was also the Laternula bivalve in there as well. So basically what should happen is that when you add organic matter to the seafloor, you would see changes in dissolved oxygen concentration, so we monitor that. And the bacteria also utilize organic matter, and they recycle it and turn it into inorganic nutrients. And that's what algae use as basically a source of their own nutrition. So you have, if algae have sunlight and nutrients, they can perform primary production. So we're examining all these interactions about the rain of detrital food, this under ice algae that's drifting to the seabed as the sea ice breaks up, and examining how that transfers through the food chain. So the animals are eating it, and that kind of thing. So
cool.